So my project came towards the end of the video, where we are trying to see how well people are paying attention, and through a game where uh, participants can control something on a visual screen through their mental effort, specifically how well they pay attention. We try to make them practice and improve their attention levels. So that paradigm is called neurofeedback. So we use EEG to acquire brain signals and analyze to see how well their attention level is. And based on their attention level, we give them some feedback on the screen. Using this, we try to help them to improve their attention and uh, try to find alternate methods to medication or therapy. So if you have any further questions, you can ask me in detail. But I guess others can also pitch in about their project.
Raj or Shankadeep, you can go ahead and talk about your experiment.
Okay, I'll go ahead because I hope I'm audible.
Yeah.
Yeah, okay. So in the project that was described in the video, so we are trying to look at different parts of the brain that gets highlighted or light up because of different kind of visual stimuli. And secondly, can we reconstruct those visual stimuli that the participant is looking under the scanner? And for this, we are using magnetic resonance imaging, MRI, specifically functional magnetic resonance imaging, where we capture a static screenshot of human brain when the participant is under the scanner. Then when they are doing a task, suppose seeing some houses or faces, then different parts of the brain lights up. And we saw in the video how and which parts they are, which lights up. And following that, we do a reconstruction problem, what they are viewing, actually, what the participant is viewing. So that is the broad umbrella of project that is being done. So if you have any questions regarding that, you can definitely ask me.
So I think there's one question from Isha. Isha, if you'd like to ask the question, you can unmute yourself. Otherwise, I'll ask the question for you. Okay, I think Isha's mic is not working, so I'll just ask the question on her behalf. We talked about the FFA and PPA activity, that how the FFA specifically lights up when faces are viewed and PPA lights up when other objects are viewed. How does this exactly happen?
Isha, different parts of the brain are responsible for selecting different kind of visual images. So whenever the information, which are the light rays from the object, enter our eyes, that goes to that goes through the visual pathway, which is the thalamus, and reach the earlier visual region, which is called as primary visual cortex at the back of our brain, the occipital lobe. And after that, it goes further into the brain. Now, since we showed you that different parts of the brain gets light up, those neurons are selective for different kind of images. So that's why different parts of the brain lights up. By lights up, we mean that those neurons are responding more than other neurons. So there are definitely a lot of neurons which are responding to a particular image, but some neurons prefer certain kind of images. I hope that answers your question.
Thanks for your question, Isha. There's one more question from Kasturi. Kasturi, if you'd like to ask.
Abhishesh, I had a question regarding the feedback. So, I imagine that this feedback helps to relieve diseases. And so, how long does the impact of this stay after a training session?
I didn't get the full question. I think there was some disconnection happening from my end.
Anushka, could you ask my question if it was not clear?
Yeah. So Kasturi's question was that regarding the feedback experiment, how long can we expect the impact of the feedback to last? And she also mentioned that it's used in some disease treatments as well. Could you speak about that?
So we can't say because it's like practice. So if you are starting to practice in a game, the learning curve could be steep, and we might see a lot of improvements initially, but uh, it depends on how many sessions we are having, at which stage the participant is, and uh, how well is he attending before the feedback even is given to them. So uh, but in the attention learning experiment, they have seen that it lasts for maybe 1 or 2 hours with one session.
Okay, thanks.
Right. There's one more question from Yugang. Would you like to ask your question, Yugang?
Yeah, hi everyone. So my question was basically that whether these non-invasive methods can be deployed for precision medicine pertaining to neural disorders as a possibility in future, or can they be deployed for diagnostic purposes only? Like, what's the future prospects and possibilities with this technology?
I think any one of you can take the question.
So TMS is actually currently also used in to uh, to as a method to uh, cure maybe depressive disorders. If I'm not uh, inaccurate, but uh, uh, so what do you mean by precision medicines here?
Like, can we use the non-invasive technologies to specifically target particular regions of the brains that have been identified being linked to particular disease or disorders?
That's right. As Vishesh suggested, TMS is one of the techniques to target specific regions in the brain and stimulate them. And hopefully, it can be used for not only diagnostic purposes, but also uh, correcting some kind of neural disorder. At the same time, there are the different techniques that we talk about, all the non-invasive technique. There was also we also discussed about neurofeedback, and neurofeedback is one area where people are working already to improve some kind of cognitive abilities. And if these tests are successful, and hopefully in the future we probably be able to at least push some boundaries in the medication. But again, this is more on a hypothesis that these things can be done or not.
Okay, thank you so much.
There's another question from Devangshi. You can go ahead and ask your question, Devangshi. Okay, I think, yeah, sure. So Devangshi has asked, in TMS, how can you measure the RMT?
So RMT is a resting motor threshold. Basically, in TMS, first we identify the motor region of the brain. It's slightly inverted omega-shaped lobe in the brain, and we point our coil to that. Then we slowly increase the amplitude of the magnetic stimulation, and we have placed the electrodes on the fingers of the subject, as you saw in the video. So on the screen, we see the EMG signals from their fingers, and uh, basically we take some set of readings, and for a particular amplitude, when it reaches above threshold, then we determine it as the resting motor threshold.
Okay, if there are any other questions, you can unmute yourselves and go ahead and ask.
Hello. Hi, can you hear me?
Yeah, go ahead.
Yeah. I want to know how likely is it that AI will be able to read our minds, like by reading our thoughts?
It's obviously a very interesting hypothesis to pose, but I don't think we are there yet. And it'll take us quite some time to get there, because there are multiple challenges involved in this particular sort of idea that we have. So first of all, we need things to be able to capture our brain signals effectively wherever we are, right? Currently, as you saw in the video, we have specialized equipments present on the brain and stuff. So we have a scanner, you have to go inside that and all of that. So one sort of progress we need to make is towards that. Can we get reliable brain signals from anyone, anywhere, right? That's one. And second thing is then, how do we, even train a model, train a AI model which can do something like that, right? So that would involve, trying to understand how to encode things like thoughts in a numerical sense, because after all, current machine learning algorithms use numbers to train themselves, right? So then what, how to encode them, and then how to bring the relation into it. And it's not that and see, though we all have effectively the same brain, but we are all different personalities. So we need to be able to capture that specific thought processes which people have. And so these are the challenges that we have, and we are not even, at least in my humble opinion, not even there for about 10% also. So it's going to take some time.
But I would also add that has been considerable advances regarding decoding of what you are trying to speak, or what you're viewing on a screen. Like suppose you're viewing a video, then people are actually decoding now what video you are seeing, or so with the advent of these language models, I think we will reach there someday, hopefully.
Right. Thank you. I also want to know if it is possible to modulate brain regions after viewing live activity in an fMRI.
So
By modulating, yeah, go ahead, Vishesh.
So the example which we showed, we used EEG signals for the participants to modulate their brain activity. The advantage with EEG signals is that it has a high temporal resolution, so you get instantaneous feedback of what is happening in the brain. But with fMRI, the activity buildup is slightly slow, so there might be some lag with what you're viewing as your brain signal and what is actually happening. But for some specific tasks, it is there. Like studies have shown that it is very much possible to modulate your brain signals.
Okay, thank you.
Priyanka, do you want to say something? You're not audible, Priyanka.
Okay, am I audible now?
Yeah.
Yeah, so I was like, okay, I'll just add to what Vishesh was saying that people are also now using EEG and MRI together, and that can help with, addressing the temporal resolution, how fast we can acquire data. Part of it, along with having, very great spatial resolution, so we can know exactly which parts of the brain are responding. So something, this combination can really, make this neurofeedback even more effective. And other than neurofeedback, there are studies with non-invasive stimulation, which we see that, it has helped improve memory in the older patient group considerably and for considerable amounts of time. So there's definite potential for both of these modalities.
Okay, thank you. There's one more question from Udaykant. Would you like to ask, or shall I read it out? Okay, I think I'll just read it out. Other than human model systems, does CPHMS use rodent models to understand behavioral patterns? So, first of all, we are not a research center. We are a science policy and think tank center. So, what we do is we promote and educate people about the use of non-animal methods. So, we ourselves do not do any research. Yes, rodent models are used to understand behavioral patterns in several neuroscience and cognitive labs. But what we aim to do is uh, we hope to reduce the use of animal models because they are not completely the same. You won't get completely accurate results as compared to a human model, especially when it comes to cognition, because a lot of the complexities and intricacies of the human brain might not be accurately captured in a rodent model. So, although they have, lended us a lot of information about developmental biology and brain development and many cognitive processes, we hope to see more models that focus on that are human based, and of course like we learned today, even non-invasive techniques can help us understand. So if anyone else would like to add on to that, please go ahead.
Yeah, I was wondering because it is CPHMS that promotes these activities, but then, of course, researchers like members from the cognition lab may be having some of their own opinions. So perhaps we could discuss this here. Do they have an opinion about using rodents? And I know that other cognition labs possibly use rodents. So has there been a consensus about whether other models can also help us or not?
I guess we could start off as thinking as cognition is it's a spectrum, right? So it's
Right.
not only lower order or higher order cognition that, cognition itself sort of underlies. And I think it's important to then distinguish for which question, which model system would be best. So it's not like rodents cannot answer any question in cognition. I think that's probably not the view that at least I would hold. But it's about what kind of cognition you want to study in what model system. So in cognition lab, we are primarily, human behavior and, models based. And that's because the questions we are asking are, probably best studied in humans. So, I mean, you could think of it as both ways that, we're also doing research which is perhaps more relevant for humans, but it's also because the research that we do is better studied in humans. I'm not sure if that distinction was clear, but it'll be nice to hear others' thoughts.
So one thing I would like to add on to that in terms of machine learning side of things. So let's say, I mean, I've seen this again and again that because so ML models are only as good as your data, right? So if you have like very good data, then it's likely that you will be able to build a very good model which explains what you have found. Now, in terms of in for example in rodents and in other animal models, it is quite possible to get neural level data with us, right? So at the level of single neurons, whereas in humans, it's not possible, and at least in our lab, we don't do that sort of research. So what ends up happening is, let's say you have a idea for a model, right? For example, let's say some new processing method that you want to try. So at least in terms of machine learning, what we can do is we can look at that the same model being applied on a rodent data set and so on, and then see how it's getting applied on the human data. So that will give you a sense of, okay, whether the issue or whether the problem is in the data or in the way that you are processing it. Basically your model assumptions are the issue. So that is one distinction I would like to also bring. So human level data, at least the techniques that we have are basically non-invasive, so they are a bit more noisy. So you might require much more stronger assumptions or stronger models to be able to get good, reliable results or outputs. Whereas, simpler, invasive techniques, because they have lower noise, it's possible that you can get a better result with a simpler model. So this also ties into this sort of pro con sort of situation.
So I think someone has mentioned, I read that Tiffer in India uses Drosophila brain for cognition in their neuroscience lab. Anyone has any comments on that?
Yeah, actually, I do believe that Drosophila is becoming, more popular in neuroscience. I had gone to a conference where a lot of the studies were from like a Drosophila perspective, and I believe that they do use it for locomotion, and I'm not sure about what other behaviors, but I've definitely heard about their flying behavior and how that can how that changes along with some interventions and things like that.
So adding on to your question, on your comment, Priyanka, I had a question on you all will be going to conferences regularly about cognition and neuroscience. So how often do you see models like the things that you're working on, like rather than using animal models to study all these things? Do you see these kind of models quite rarely, or are they coming up more? What's the frequency of or the number of people who are using these types of things?
So I'd say especially in systems neuroscience, like cognitive and behavioral science, there are a lot of human studies. And that's also because technology has gotten better. So we have better neuroimaging, and we have, neurostimulation and just pure behavior too. But along with like humans, there are a lot of non-human primate studies. So I'd say those two are the like sort of the mainstay of at least this side of neuroscience, where there's a lot of monkey behavior and recordings and some stimulation too, but at a more invasive sort of way. And maybe some other lab members might want to add.
So I think it depends on the conference we are visiting. So as Priyanka had earlier suggested that there are what kind of questions we are asking. So some conferences may cater to very specific questions which are related to human behavior. And since I have visited to such conferences, so those conferences there's more focus on using the kind of model that we are using for different kind of studies of cognition or languages to understand human behavior. But alongside that, there are conferences which are more focused towards molecular side of neuroscience or behavior in animal models. So I think they might actually be using animal models to do their research. But I have not attended such conferences yet.
But in general, in the department at IISc, we have like a whole range of labs. I mean, there are labs which use mice, they use rats, and then you have, monkeys and humans. And also like the petri dish. I mean, there is a lot of stuff even at that level. So the whole range, if you consider just neuroscience.
Right. Naina, you can, if you'd like to ask your question, you can go ahead. Otherwise, I can ask it for you. Okay, I think I'll just go ahead. Quick, no, she's unmuted. Go ahead, Naina, please. You're not audible. I suppose I'll just ask it on her behalf. There are in vitro human organoids being created in the lab these days, which are used in disease modeling. Can these models be practically used here to learn behavioral patterns of humans? Any one of you can answer the question.
So I'll probably first start off by saying that we do not have expertise in this particular field. Neuroscience, at least the one that that we are more familiar with, uses animal models even of disease, right? So suppose we consider Alzheimer's, there are many animal models like mice models of Alzheimer's, and then people have studied disease progression and how medicines affect the other stuff like that. But I'm actually not so sure about in vitro human organoids being used to, at some point study human behavior. But I believe that there is some sort of effort in that area where in a petri dish, people are like, they've sort of cultured neurons and they've trained them to play a game. So I believe there is some effort in that area, but it's not mainstream yet.
Thanks for that answer, and thank you for the question. If anyone has any more questions, please go ahead. Otherwise, we'll wrap things up. So I think that's all. Thank you everyone for, oh, there's one more question. Is there free software for cognition analysis about Drosophila or zebrafish? I don't know if you guys know about this, but
I think it depends on what kind of analysis you want to do, like what kind of data you have and what do you want out of the data. So like you can always find Python scripts and analyze the data to get some results. Python libraries are totally free.
okay. If you guys would just like to close talking about the work that goes on in your lab and you guys also have open spots for participants. If you'd like to talk about that, please go ahead. Can you hear me? Was I audible?
Yes, you were audible. I think we were trying to understand who's going to answer first.
So our lab uses, like, our lab studies humans, and we are always looking out for participants to collect data and do our tasks so that we can study their brains. So if you are healthy and above 18 years old, you are welcome to come to our CNS and take part and see what is happening in the lab.
All right. So I think we'll wrap this up. Thank you everyone for joining us. We hope you had fun watching the video. Very sorry for the technical issues, but the video is up on YouTube, so you can go watch it there, and you can check the chat box for links to the workbook for episode 4, the assignment link, and also a feedback link, so you can let us know how the video was made and if you have any suggestions or any comments. So thanks for joining us again, everyone, and we'll see you soon.
Thank you, and also big thank you to Team CPHMS, because this was very fun exercise even for us, and great questions. Yeah, we had fun too. Thank you.
Thanks.
Yes, thank you.
Thank you.
Bye, everyone.